Chapter 20. We're going to be dealing with antimicrobial drugs. I chose the quote for this one since we have Alexander Fleming on I chose him since he was the guy that discovered penicillin. The unprepared mind cannot see the outstretched hand of opportunity. Every time you get a chance to learn something new, it's an opportunity to expand yourself and improve your okay, so we want to help you guys learn. Alright, so very important for pharmacology, very important when you're dealing with patients who have bacterial infections. Okay, alright, so we're going to be talking about a variety of things, and we're going to get into basically starting off. Now, when we think about drugs and chemotherapy, we think cancer. But chemotherapy is this use of drugs to treat disease. It is chemotherapy for treating cancer is the common that we always hear. But Ehrlich developing salvarsan. I know some say salvarsan. The salvarsan is the way I've always heard it said. But anyway, this is an arsenic compound to treat syphilis in the early 20th century there. That was the first true chemotherapeutic agent. Antimicrobial drugs, these interfere with growth of microbes in a host. They can do a variety of things. They can mess up the replication, the transcription, translation, mess with the ribosomes, for example. They can mess with the enzymes of replication and do a variety of things. Antimicrobial drugs, one we have is an antibiotic. They are made by microbes. They are made by other microscopic organisms, whether it is a fungus or a bacteria, or what have you. They make these in small amounts to inhibit other microbes. More than half of the antibiotics are actually made by some kind of Streptomyces species. Drugs that kill harmful microbes without damaging the host, the so-called magic bullet, is got selective toxicity. So one of the things, for example, if I want to give myself things like penicillin to affect microbes, I can kill them, I can kill the microbe. Now, if I wanted to take something for like affecting worms, the things that affect worms can also harm you. If it affects other eukaryotes, it can hurt you as a eukaryote. So selective toxicity, the magic bullet, that's what we found, the magic bullet with penicillin. That's how it was a magic bullet. And I always like to kind of mention that so-called magic bullet idea, that it was harmful to microbes but not us. Okay. Now, 1928, don't worry about the year, but I definitely know that Fleming discovered penicillin. I may ask it again. I know I've already asked it once before. Mold contaminated petri dish. So this was basically a happy accident. We found that the mold that actually, the penicillin colony that actually got on there, inhibited the growth of bacteria. And then, so it was 1928 when he discovered it. 1940, it finally got clinical trials and moved. It went from the UK to US during World War II. I have an aunt and an uncle who both died of strep throat because they were born before penicillin was available, and they got strep throat and died from it. My dad was the youngest of 13 kids. He was born in 42. So these kids were born before you could actually get penicillin here. Okay. Now, antibiotics, they can have really kind of two. There's what we call narrow spectrum and broad spectrum. Narrow spectrum may affect gram positives, but very few gram negatives. Broad spectrum affect a broad range of gram positives, and they affect gram negatives. We usually give a broad spectrum antibiotic if we don't know what we're fighting, if we don't know what the pathogen is yet, and we want to go ahead and get it in there. Bacteria are the easiest to kill with antibiotics, and we are looking for new ones. If it is bactericidal, remember cidal kills, static prevents growth. Bactericidal stuff kills the microbes directly by doing a variety of things, like messing up gene expression or messing up replication or growth or something like that. Bacteriostatic just prevent them from growing, prevent them from spreading. All right. So microbial, one of the big things they do, which I have talked about a little bit earlier, is inhibiting the cell wall synthesis. One of the things I do want you guys to know is what antibiotics do, especially specific antibiotics when mentioned, what they do. Penicillin weakens the peptidoglycan. Basically, it affects transglycolation, transpeptidation of the peptidoglycan of the cell wall, making the cell wall weak, making the cell lyse, especially forming those we talk about like L forms and things like that. Inhibiting protein synthesis, prokaryotic ribosomes, they are different, so we can affect antibiotics to go to the prokaryotic ribosomes, the 70S ribosomes, so we can inhibit protein synthesis. We can inhibit nucleic acid synthesis, so it just can't make genetic material. We can interfere with DNA replication and transcription of DNA by affecting the enzymes involved. We can injure the plasma membrane, like actually change the permeability with a sterol can be disrupted, or loss of important metabolites that are important in the cell membrane, injuring it. We can inhibit the synthesis of essential metabolites through competitive inhibition. Use a competitive inhibitor, use an enzyme inhibitor to affect the bacteria. And hopefully we can use those, still have those at work. I have seen things from some of the best microbiologists saying that I have about 50 years before antibiotics do nothing, unless we develop something new and understand. Like, one of the things they're saying is we've got to understand how bacteria make us sick. Okay? All right. So we can affect the DNA, we can affect transcription, we can affect translation, we can affect replication, we can affect DNA synthesis, nucleic acid replication, transcription, things like your quinolones, like ciprofloxacin, chloro and fluoroquinolones, things like that, and enzyme activity can be affected, just things like that. We can use, for example, penicillins for cell wall, penicillins, cephalosporins, bacitracin like you find in neosporin, vancomycin, those kinds of things. We can inhibit protein synthesis, the chloramphenicol, erythromycin, the tetracyclines, streptomycin. We can inhibit the synthesis of essential metabolites like sulfonamides and trimethylporine or trimethylthorpin. Sorry, sometimes I haven't heard these in a while, so it can be a little hard for me to say. We can injure the plasma membrane, polymyxin B, things like that. Okay. So anyway, I chose to have a picture of these. I want to talk about penicillin, what we call these called beta-lactam rings. Here we have this little square right here. This is called a beta-lactam. This little square. They prevent the cross-linking of peptidoglycan, making it weak. There's about 50 related antibiotics. Their core structure is a beta-lactam ring called the nucleus of it. There's natural penicillin from penicillium. There is beta-lactamase made by gram-negative bacteria, the enzyme that cleaves the beta-lactam rings. A lot of your gram-negatives make beta-lactamase. There is synthetic, semi-synthetic penicillin made in the lab, resistant to that. It's broader spectrum. One other thing we can do with a lot of these beta-lactams is they work in the same way for cell wall synthesis. Definitely penicillin, you need to know the penicillins, what they do. I won't give you a picture of penicillin and ask you guys what is it, okay? Now, vancomycin is used to kill penicillinase-producing streptococcus, or as I like to call them, beta-lactamase. That's how I'm used to saying it. That's how I learned it. They are the last line against antibiotic-resistant Staphylococcus aureus. We do it IV. It can be toxic to ears and kidneys, so it is last line of defense. It is a last line. Amoxicillin is common for children. We add clavulinic acid, which is beta-lactamase inhibitor. We take it with amoxicillin, and it's called augmentin. It's made from a Streptomyces clavigarius, clavulinic acid made from a Streptomyces species. Cephalosporins are used against penicillin-resistant strains, and bacitracin is topical against gram-positive. So what we found in Neosporin is bacitracin. Know these different antibiotics. I'm going to ask you antibiotics, okay? So cell wall mycobacteria are different. They have mycolic acids, which are waxy. They stain with acid fast, so we might use an isoniazid. They inhibit mycolic acid synthesis. Good for Mycobacterium tuberculosis. Ethambutol inhibits the incorporation of mycolic acids in cell wall. So again, we can do cell wall for other bacteria, like tuberculosis is one that I want to mention. Tuberculosis was a death sentence for people as well. If you guys played Red Dead Redemption 2, tuberculosis that killed Arthur. Okay. Or famously what was had by Doc Holliday. Okay. All right. We can affect protein synthesis. We can prevent synthesis of 70S ribosomes with chloroamphephenical. This prevents peptide bond formation. Aminoglycosides, streptomycins produce. They change the shape of the 30S portion of the ribosome, so you don't read the mRNA correctly and you end up messing things up. Tetracyclines, they actually mess up how the tRNA attaches to mRNA. This is why I had you learn tRNA. They prevent the addition of amino acids. We can prevent the formation of 70S ribosomes with oxazolonidones. They bind where the 50S would be normally bind to the 30S, so they prevent the binding, the large and small subunits from coming together. So there's all kinds of things we can do. We can change the shape with streptomycins, tetracyclines, things like that. We can really mess up the ribosomes and affect translation, inhibiting protein synthesis. Alright, now we can inhibit nucleic acid synthesis with rifamycin, inhibits RNA synthesis. We can use that to affect tuberculosis. Quinolones, fluoroquinolones, DNA gyrase, for example, which no replication fork, things like that. Replication is messed up. We can injure the plasma membrane with polymyxin B, makes the membrane holy. Isoniazid, and we can inhibit the fatty acids in the membranes, like in tuberculosis as well. Sulfonamides, they are competitively inhibit what is called PABA, para-aminobenzoic acid, and that prevents it from being turned into dihydrofluoric acid to prevent folic acid from being made, which is an important nutrient for organisms. And we don't use that metabolic pathway to make ours. TMP-SMZ, trimethoprim, and sulfonamethoprim. My brain sometimes has a hard time saying these. Your sulfamethoxazole. I'm going to pause for a second and say it. Sulfamethoxazole. Sorry. It can be really hard to say those, and I practice them all the time so I can say them. And that actually competitively hits dihydrofluoric acid as well. Dihydrofolic acid synthesis, again, prevents folic acid synthesis and things like that. So it prevents essential metabolites. TMP-SMZ is what you will hear it called. Anyway, sorry, I now can go. Polyenes, they combine with sterols and plasma membranes, so they're fungicides. These are antifungal drugs. Azoles and alamines. These are where I'm a little bit like I didn't get as much training when it came to antifungal stuff, but the azoles and the alamines, They interfere with sterol synthesis, used to treat skin infections, mycoses like ringworm, jock itch, athlete's foot, yeast infections. So they're usually found in lotions or creams. Echinocandinins interfere with fungal wall synthesis. They lyse because they don't produce the cell wall properly. Griseofulvin interrupts cell division. It's used to treat ringworm, like a skin infection. Ringworm is not caused by a worm; it's a fungal infection. Antiviral drugs. Viruses replicate inside, so we can use genetic and metabolic mechanisms of the cell against them. It's hard to target without targeting the cell, which can also accelerate mutation rates. One of the things we use is interferon. Interferon is produced by our cells to help prevent the spread of viruses. Proteins are made by infecting host cells. They inhibit the spread of a virus to new cells, so we can make interferons as a treatment. HIV and RNA-based virus depends on what is called reverse transcriptase to actually control RNA synthesis. We can use an antiretroviral, a drug to treat HIV, and block the synthesis of RNA from DNA to prevent viral replication, something we can easily treat. There's lots of antiprotozoan drugs. Quinine is an old one to treat malaria, but they mess up anaerobic metabolism. Quinacrine to treat Giardia lamblia, things like that. Chloroquine is replaced quinine. Quinine was made by a yew tree, I do believe. Antihelminthic drugs. These are also hard to treat because they're eukaryotes. They treat helminths, worms, parasitic worms. Niclosamide kills tapeworms by inhibiting ATP synthesis. If you can affect a eukaryote, you can affect us. Parsaquintel kills tapeworms by altering the permeability of the plasma membrane. Mebendazole inhibit the formation of microtubules in the cytoplasm, so you can't do a variety of endocytosis. They can't do endocytosis and get things in. There is loads of resistance to antimicrobial drugs. Antibiotics for bacteria have been resistant, a disease for bacteria. What basically happens is you have an antibiotic for a disease. We're treating the disease, but the bacteria is what's causing it. We have our first exposure of it. We give it susceptibility. There's a really high mortality rate. The bacteria is susceptible to the antibiotic. Only a few survive. The survivors have genetic characteristics that account for the survival. The progeny are now more resistant. They reproduce. Now those genetic differences are more out there, along with random mutations and plasmids like R factors will make them even more resistant, and then resistance happens. Okay. There's many things that can happen. That's basically how you give an antibiotic. The first exposure, it wipes out a lot of bacteria. Some survive. The progeny are now resistant, just like the survivors were. And those genetic differences continue on with random mutations, and it gets worse. Now, resistance to antibiotics, one of the things we can do is just not let the antibiotic in. We can modify the porin proteins that are found in the cell wall. We can prevent the entry of the antibiotic. You're resistant to an antibiotic if the antibiotic can't come in. We can destroy or inactivate it, like what happens with the beta-lactamase enzymes that deactivate the beta-lactams. We can alter the target molecule. We can actually modify protein synthesis at the site of mRNA, and it just doesn't work anymore. We can pump out and eliminate the antibiotic. We can eject it, efflux pumps. So bacteria can efflux it. So we have an antibiotic. We can block it from entering. We can target it. We can use an enzyme to alter the antibiotic, make it no longer antibiotic. Enzymes can inactivate the antibiotic. We can pump them out. We can do a variety of things that make bacteria resistant. So basically, we start antibacterial therapy. The antibiotic is started. We kill off the bacteria. Then antibiotic resistance happens, and then these we get back to a higher level of population, and we have to change antibiotics, change treatment, use multiple antibiotics, and things like that. There's a lot of things we can do to help prevent this. One of the things is misuse. Bacterial antibiotic misuse is a really big thing that leads to antibiotic resistance. This can be using outdated or weakened antibiotics. Old antibiotics that have been sitting, stored incorrectly, taking old antibiotics. Don't do that. Don't take antibiotics for viruses, antibiotics for a cold, or other inappropriate conditions like COVID. Using antibiotics in feed, that's one big thing, is antibiotics go in animal feed. Next thing you know, the animals are constantly using antibiotics. They're constantly being treated with antibiotics. And then bacteria in a farm become antibiotic resistant. Then they cross over to humans. And then we get antibiotic resistant bacteria. Failing to complete your prescribed regimen. So if you need to complete the entire bottle, using someone else's leftover prescription, you didn't take all of it, they didn't take all of it, and you weren't taking the whole regimen. You need to take the whole regimen, okay? You're contributing to antibiotic resistance. Now, combination of drugs may be something we have to do, okay? Like amoxicillin and clavulanic acid. We may need to use another drug with it, okay? Synergism is two drugs together greater than the effects of those alone. Synergistically, the two drugs, you take one drug by itself, it doesn't work. The other drug by itself, it doesn't work very well. Then you put them together and it is much more effective. Okay? Antagonism, the effects of the two drugs together, is less than the effects of them alone. So some drugs inhibit the effects of each other. Okay? Drugs do work together really well. Sometimes we have to try two drugs at one time for a synergistic effect. There are lots of things. This is what some of my research was in, is antimicrobial peptides from the skin of amphibians. I worked on an antibiotic peptide, antimicrobial peptide found in the skin of spotted salamanders that worked as an ATP synthase inhibitor. It not only did it take E. coli's ATP synthase inhibit it, but it made it run backwards, made it ATP degradase. Bactericidal and broad spectrum activity, part of the defense system of the host, so not harmful to us, but potentially could affect cancer cells. This is one of them is magainins from amphibian skin glands. Niacin, lactic acid bacteria produce it. Cecropin by a silk moth. Melittin is it's found in a bee venom, and defensins, which are made by many animals found in neutrophils and white blood cells. Insects, plants, birds, and mammals all apparently have defensins, okay, for their help fight diseases. There are antisense agents. They synthetic strands of DNA. They block the biosynthesis of bacterial toxins. The nubiotics, so called, involve nucleic acids. RNA interference. We can actually stop gene expression being transcribed into what is called an siRNA. To make double-stranded, that's used to make a double-stranded RNA in virally infected cells. Small interfering RNAs, drugs that can be repeatedly prevent protein biosynthesis. They can bind to mRNAs, making it double-stranded, and it can't be read anymore. So basically what these siRNAs do is temporarily make double-stranded RNAs, and it can't be read, and they can be destroyed. Small interfering RNAs. Alright, so I hope you learned a lot about antibiotics. That is it, and I will see you in the next one.